I would like to get started with a quick self-introduction. So with that, I don't know, do you want to get started?
Sure. Hello, everyone. So my name is Aydogan. I'm a faculty at UCLA for the last 17 years. And I've been working at the intersection of optics, photonics, microscopy, sensing, and machine learning, AI recently. And started up a few companies, and the latest being Pictor, which in Latin means painter. So we're painting microstructure of tissue. That's how I opened up every single pitch that I had during COVID.
So before we came on stage, I learned something new about you and your lab. So I know it's off topic, but I think it's important because it's part of our mission of service and education. Can you just, in 30 seconds, describe what you're doing with undergrads?
So every fall, we recruit to our lab 30 undergraduate researchers from UCLA across 10 different departments. They do hands-on research and publish with us. In fact, even for a cutting-edge research company like Pictor, some of those undergraduates contributed to the IP and joined Pictor. So, and this has been continuing for more than a decade. At the peak before COVID butchered it, I had 63 undergraduates at the lab doing research and literally hands-on research, not just reading papers.
Great. Yair, tell us about yourself.
Good to be here. Good to be invited. By the way, I was a mentor in one of those programs. Again, it's off topic, but there was a year that I actually was mentoring 7 of those undergrads at the same time. Yeah, that was the 63 cohort. So great experience for both myself and hopefully for the undergrads as well. So I think that was the year that we came up with the IP that we end up spinning out to Pictor Labs. So again, Pictor Labs was born here in UCLA on the 5th floor of the building there behind us. So it started as a very interesting idea. We were basically, it was the early days of, the new renaissance of deep learning. And, we were exploring all sorts of very interesting ideas, both of us coming from a background of microscopy and imaging and computational imaging. And, we're exploring all these amazing ideas on, how can we increase resolution? How can we, get more information out of each and every microscopy image? And again, everything is like through microscopy. That was our lens, as they call it, or a prism. And then, we basically came up with the IP because we were working a lot with, back in the day, pathology slides, and came up with the IP. And, right now it's a company with 25 people. Again, we spun out from UCLA after 2 rounds of funding, a Series Seed and a Pre-A, raised almost 19 million. So I think, for me, the gist of it is, when you start your postdoc, you never know where your career can end up. Like, I think it's one of the great things that an environment like UCLA allows you to do, and to develop yourself and your skills, and then take it out to the world and spin it out to a company like this.
So, you mentioned microscopy and pathology, but I think that for some people in this audience, it's still not very clear what is your technology. So, Aydogan, perhaps you can take us into a semi-deep dive into the technology and what problem you're solving.
Sure. So how many of you gave a biopsy so far? So every time that biopsy is essentially sectioned into thin layers, microscopically thin layers, and for the last century plus, they're stained because diagnosticians need contrast to see subcellular features, etc., to diagnose all kinds of conditions. We do the staining using AI without any chemistry, without any histology processing of those thin sections. We can stain label-free tissue as if, from the pathologist's perspective, it's coming from a histology lab. Advantages: faster, cheaper, toxic chemical free. You can do multiplexing, and you can take more out of your small biopsy, which means you preserve tissue for other things of interest, and a panel of stains that you want can be done in minutes using GPUs that look at microscopic images of the same tissue in a different contrast mechanism that is transformed into what diagnosticians are accustomed to seeing digitally, virtually.
So, earlier today, we had several talks discussing about applications of AI in medicine. And one of the recurrent themes were that you need to get the right diversified data set in order to get the right results that are not discriminating. How do you attack this problem?
That's a great point. So Yair can tell you today at Pictor how long does it take for us to create one stain from scratch, the efficiency of the process. He can tell you more about how we've streamlined the process. But going back to this question, microscopy and virtual staining of tissue chemistry-free is extremely efficient because a single tissue slide, whole slide, contains billions of pixels for us. That if you look at the ground truth coming from histology, the ground truth is at the diffraction limit, 200, 300 nanometer, that pixel. And I have billions of those, which means with approximately 50 cases, I have tens, hundreds of billions of ground truth that I learned from. Since my end goal is not to diagnose the patient, but create multiplex virtual stains, chemistry-free, I don't need thousands of patients. And that's essentially a very interesting secret sauce that we have at Pictor. Maybe Yair can tell you more about what we've done to kind of streamline it for any stain tissue combination.
Right. So I think it's very important. One of the things that you want to do with an AI product as soon as you can is to build the right pipeline. And the right pipeline is, first of all, of course, there is the data and how do I get data? How do I procure data? But that's just one end of the equation. The other part is, I'm sure, if you're all from LA, or most of you, you've seen those Waymo cars running around. I don't know how many sensors those cars have. I'm sure they have at least 15. That's kind of what I'm counting. So let's try to imagine that. They're running around for hours. They have 15 sensors. That's a lot of data that needs to be ingested. And then, we need to figure out what to do with it. So I'm sure there's a lot of people in Waymo that spent a lot of time in building that type of a data engine. How do you ingest the data correctly? How do you find the interesting features of it? And then how do you take it and use it for your training? So that's, I think, one of the most important things when you build an AI product. And this is something we put a lot of emphasis in Pictor. So now we can, get those models from 50 slides. Once you get the 50 slides, you get those models in a matter of 3 to 4 weeks. You can start getting those stains running around.
And here, how do you avoid the AI hallucinations?
So AI hallucinations. So first of all, it's an interesting question. I mean, in terms of we're training this versus a ground truth, okay, that is coming from those hundreds of billions of data points, as Aydogan mentioned. So I think that's one of the key points that we have here. And the second thing is we basically tailor all sorts of specific loss functions in order to avoid any type of hallucination. Number 3, we always have human in the loop that, first of all, helps us select the data that goes to training, but then also supervises the results on the other end of it.
I can add something on it. So there are 2 kinds of hallucination when it comes to pathology. One of them is the hallucination that is not going to mislead the diagnostician. This happens all the time in the chemical workflow. Flipping of tissue, tearing of tissue. These are mistakes of the clinical workflow due to the chemistry. They will never mislead the diagnostician, and that's the standard of care, right? So we can make those kinds of hallucinations. That's perfectly fine. Sometimes it happens to us. We, for example, for a stain, there is no green color. Our network inferred green color because at the tissue there was a dust particle. When we were imaging, we realized later there was a dust particle that was autofluorescing, which is our bread and butter. And it revealed our network to hallucinate green on a stain that there was no green. That is a good hallucination. It's a physical hallucination. It will never mislead the diagnostician. We're perfectly fine. We shouldn't imagine or hallucinate new patients because nobody wants to pay taxpayer money to diagnose patients that never existed. And that's the one that we're protecting the models not to do. And there are different ways of doing it. In fact, you can even train additional AI as a watchdog to look at inference. And this is something that is going to have a new UCLA IP on it soon.
I like it.
Yeah, there's going to be new UCLA IP on it, a watchdog for hallucinations in pathology. I'll email you soon.
So let's move on from the technical side to the business side. And the first thing that any company needs to do is fundraise. And you already raised $80 million. That's quite an achievement. Congratulations. But let's speak about the early days when you just got started. How did you get your first $1 million or $5 million?
So the story actually is from an academic point of view, it's a little bit interesting. How many of you are faculty or researchers at UCLA trying to kind of spin off companies or already spun off companies trying to do it again? So this might relate a little bit more to you. The story starts actually in 2018, where we published a science paper on shaping materials for all optical computing, so diffractive computing, nothing to do with pathology. But that science paper attracted a lot of VCs to knock on our door and saying, hey, let's AI is big, power consumption is big, let's use your structured materials for computing, right? At that same time, we published our first Nature Biomedical Engineering paper on virtual staining. This was put in archive 2018, filed a patent application in 2018 before it went into archive. Publication took a year. They overlap. So as an academic, it's very tempting to say, okay, cool, people are excited about a science paper. Let's transform, computing for visual scenes. We didn't do that because the technology readiness level at that stage, you got to be honest yourself, it wasn't as high. It was a scientific exploration, the science paper. That's why it was published in Science. But the Nature Biomedical Engineering was more advanced in terms of what you can do with virtual staining and histopathology and everything. So we spent the first, literally, 6 to 9 months to convince our potential investors that they should invest on virtual tissue staining as opposed to diffractive materials, which was published in Science, and that was glamorous for them. After that stage, of course, unfortunately, COVID hit. So we were supposed to meet at UCLA to show our working systems prototype to many investors at UCLA. Everything shut down. So we had to pitch over Zoom. And that's why some of the pitches, actually, my wife could give better than me, because after a while, she memorized how I start. Pictor means painter in Latin. We paint the microstructure of tissue. I still start my talks like that because I memorized it during COVID. It was tough. 2020 was a tough year, Zoom-based.
But when we spoke before, you said something that was very interesting to me, that you didn't immediately go to the VCs, if I remember correctly.
Yes, that was interesting because I was trying to kind of digest the virtual staining technology and kind of see what to do with it. And we were actually, both of us were slightly misled. We are coming from a device-heavy, physics-heavy background, and we were thinking we're going to build our own microscope. And then it will be a microscope plus virtual staining. That was our idea. But VCs found us indirectly through another basically paper. And then we kind of started to align them and convince them on virtual staining. During that process, 2020, we also dropped the idea of developing a hardware, which would have killed us, basically, if we did that. Because pathology is a very structured and conservative field that they have their own machines there. It's a fragmented market. And having another machine there was a terrible idea, which we had for different reasons, but perhaps later.
So it's interesting to understand that there's no contradiction between high-end scientific publication, it's science, and fundraising. Actually, they're complementary.
It is actually PR is amazingly powerful. So for my academic ventures or spin-offs, I use PR as a carrot to attract people who wanted to fund me. Some of them were VCs, some of them were foundations. I had once actually from a foundation talk to me about a disease that I didn't know about. As I was talking over the phone, I was Wikipediaing kind of like what disease they're talking about. And they funded my program. And I created a lot of IP on it, by the way, over the last 10 years, which is something else.
So Yair, one of the previous panels spoke about the challenges in finding a good business model for an AI-based company. Can you expand a little bit about your Pictor's business model?
Right. So in our case, histopathology is literally on every facet of medicine that you can think of. So, it goes from, safety studies like food safety, vaccine safety, drug safety, all the way to research. There's a lot of research activity. And then, on the other end of the spectrum, you find all the clinical activity. So clinical could be, actual patient diagnostic, but it could be some other types, for example, clinical trials, etc. So it's all across the board in medicine. So in that sense, our technology is going to replace workflows in the lab. So in some senses, there are sometimes challenges, I think, for AI and like what is exactly those what is the application in terms of people that it will either replace or augment or whatever. So our technology is not replacing the clinicians. So it's all about efficiency in the workflows in the lab that is going to translate to the clinicians that are actually looking at the result and make them more productive. So if I want to kind of broaden this thing, it's all about, how much of change in the workflow your technology creates. And, I think we're lucky because in our case, we can use existing equipment, that's microscopy. We have cloud services that are already there. So that's another thing. That's another barrier that we don't need to cross. And we do not interfere with the work of, I need to say, the decision makers, which are actually the clinicians. We're kind of upstream from there. So I think, we got this very good combination. So, generally for an AI product, you kind of need to find the niche of where your technology is disruptive, maybe I should say transformative rather than disruptive, in order to make it acceptable quickly.
So you have a platform technology, right? Is there more than one market for this technology, more than just the labs? Where do you see it heading?
So most of it, again, is on these multiple verticals that I just mentioned, both in the clinics, both in all the safety studies, again, all the animal tissue, for example, diagnostic. We're talking about tens of millions of different slides and tests that are being done per year. Again, all the vaccine efficacy, for example, start with animal testing and animal studies. So, and again, all the research lab, like literally every university is spending a lot of money each year on these different tests. So there's a lot of market for those type of things. It's around 50 billion a year.
So if we divide that market, there are three categories that are very important. One of them is the preclinical market, research market. Our technology also works on animal tissue beautifully well. Pictor has a panel of all kinds of organs of different kinds of model organisms, right? On top of that, we also target laboratory developed tests. And at the top of the pyramid of opportunities, we have primary diagnosis. So the FDA landscape is, for a platform technology like this, is very convoluted, and not everything that you do is going to be FDA regulated when you do virtual staining. It all depends on who's going to use it for what. For example, if it's primary diagnosis of the patient, yeah, that's heavily FDA regulated. Most likely you're looking at a class 3 device. But if it's teleconsultation, telepathology, remote services, that's a different game. College of American Pathologists dominate that in terms of their guidelines. For LDT route, it's completely also different. So that's a pyramid of opportunities that we're trying to prioritize, given the fact that we have limited funding and we must, deliver. And one thing that I want to mention very briefly is, in pathology, currently there are two sides: digital and gloss. So, and they're fighting with each other. Gloss is the old school. I don't care about digital pathology. I look at everything on a gloss slide with my microscope and diagnose the patient and write my report. UCLA is hybrid. Most cases are signed, according to my colleagues, based on gloss, but we also have digital pathology infrastructure. We position ourselves at Pictor as a tipping point for digital pathology to eat away gloss-based pathology, because we provide so many advantages to digital pathology workflow that gloss can never do, that it's going to be end of the game, in my opinion.
Can you elaborate? One of those advantages that you mentioned before was the sustainability aspect of digital. Can you say a few words about that?
Yeah, actually, this is a funny story, because one of our investors cared about green technologies as part of their investment portfolio. They looked for green technologies, and AI doesn't shine in that list with the GPU consumption and everything. Well, we did a study on how much toxic wastewater traditional histochemistry creates, and we're saving, guess what, millions of gallons of toxic waste through virtual staining, which is offsetting by a big margin the GPU consumption and electricity, etc. So it's a green technology. It's of course, for digital pathology versus gloss-based pathology, that's not the real weapon. The real weapon is multiplexing. You can multiplex stains that gloss cannot. On top of H&E, you can do immunohistochemical stains. Yair can tell you more about all of our customers and how much they're excited about advancements in speed of staining and multiplexing that gloss-based technologies or standard care of workflow cannot do.
Yeah, so a couple of more things about the green part. So right now, really the workflow is all about getting reagents to a lab. And then, in the lab, they use these reagents to put it on these tissue sections, which create those stains and ends up being the test. So imagine a world where you don't have those reagents. You don't need them for anything anymore. You just have one box, and that box can do all your tests, and at the same fashion. There's no specific reagents for different tests, and it's all being done, again, the same manner as what was just mentioned. We can do those multiple tests on the same tissue section, so you can save a lot of those tissues. A lot of those clinical studies can be reused. Many patients could, maybe be eligible for multiple drugs. There are some scenarios where, they just cut through the entire block that they got from the biopsy, and the patient is no longer in the system in some senses, so they cannot use that information anymore. But if you have one tissue section that represents the entire blocks and the entire myriad of tests you can run on it, that's a huge advantage. So I think, going back again to the green angle, really it's all about how you take it and disrupt an entire supply chain. So, by the way, one of our investors, they one of their angles was the Suez Canal channel blockade. So they couldn't get their stains there. So the reagents that they're using for the stains. So that was kind of a tipping point for them to wait a second, this technology could resolve those type of issues. So that was another interesting data point.
Interesting. So we were 20 minutes into our conversation, and I have one last question before we open it to the floor. It took you 20 minutes to mention the FDA. That's really rare when you speak about medical device. How come? Are you not working with the FDA? Are you not seeking their approval?
Well, we are right now planning for FDA discussions this year, and Yair can maybe tell more about those. The reason that FDA did not pop up as early is, especially think of a pyramid of opportunities where the baseline is research and preclinical and maybe secondary consultation, teleconsultation services, and laboratory developed tests. Those are currently outside of the FDA regulatory body. And that means for a company like us to find our customers, to get our first prototype products in the hands of our customers, to understand the engagement with what excites them most, that's a great playground. But that also prepares us very well for the upper steps of the pyramid where it's more regulated and you will need to work with the FDA, which we're preparing.
And that market is not that small. I mean, it's by the most conservative estimation, it's around 13 billion. Some would say, closer to 20. So there's a lot of, room for us to grow and develop our product and our offering on those markets while we're, starting the discussions with the FDA. So again, you don't always have that type of advantage, but that's part of the advantage of having a platform technology in that sense. So you can, really go to the other verticals and then go to the FDA once you feel and understand what the market would really want out of your product in terms of, specific assays that we're going to go for the FDA with.
So I want to see if we have any questions from the audience. Okay, so we'll continue. Any during your journey, what was the moment that you thought things are not going as well as they should have?
Well, COVID. COVID shut down, canceled a lot of our meetings and travel with VCs. So that was, of course, that was recovered by with some delay. It cost us maybe 6 months, but that's okay. But one thing that COVID, as Yair mentioned, accelerated is we found a great partner, investor, because of supply chain issues. COVID created a lot of supply chain issues also for pathology and histology. Guess what? Perhaps 90% of those stains that are used in the clinical workflow, they're coming from one country. And when COVID hit that country, of course, very sad, but that was the tipping point for our pitches for at least some of our customers or potential investors who were literally in that business. The other aha moment perhaps was really the time that we kind of convinced ourselves that we should not build hardware, not a new microscope, just as kind of like Intel Inside. So you don't have to build a computer, right? You just provide the intelligence to it. Our current motto is the same, Pictor Inside. For if you are a microscopy company in pathology field, Philips, the first ones that went through the FDA for Class 3 process to go digital pathology, Philips scanners, Hamamatsu, Zeiss, Leica, you name it, Olympus. We want to be inside all of them. Why should I build a new microscope? Well, I built microscopes and created products out of it, right? So that's what I've been doing for 17 years at UCLA. That was kind of like my motto. I'm going to create a microscope. But that was a bad idea.
Yair, one last question. What's the next big inflection point for Pictor?
I think I would have to say that the point that we get to what is called a lab developed test. So again, the gist of it is a lab developed test is, so most stains are governed by those rules that say, okay, if your lab validated those stains based on CLIA guidelines, and they have the record for it, then you can use it. Okay, so they always compare it to, the gold standard, and then it can be used. I think for us, that's the next inflection point in terms of our product development. That will get us into the world of, of clinical samples. And I think that will be a huge inflection point for us.
Not to mention fundraising. So we're trying to double our company from about 25 to 50 and currently looking for checks.
Thank you very much. It's been a pleasure talking to you.
Thank you. Thanks.